So we left off, we were talking about the innate immune system. If you remember, this was the immune system that is non-specific, so it's going to protect you against all invaders, all infectors, if you will. And we mentioned that part of our innate defense were the first line defenses. Those being the skin and the mucous membranes that pretty much line the body proper, the antimicrobial substances that we secrete, and then our own normal microbiota that serves to protect us as well against pathogens from infecting us. We then went on and introduced the different types of white blood cells. A couple of things to keep in mind here. We identified that there were 4 main roles of the white blood cells. Either phagocytic, antibody producing, cytotoxic, and regulatory. So with every white blood cell that we come across, you should be able to apply one of those roles to that white blood cell. So we've introduced the neutrophils. The neutrophils are phagocytes. That's their main job, to go out there and eat foreign cells, foreign material. We haven't really talked much about B cells yet. We will later tonight. They're the main antibody producers. We mentioned basophils earlier. What do basophils do? They release histamines. They serve to help regulate our inflammation response. We introduced eosinophils. Eosinophils, remember, those are the granulocytes that attack big worms or big cells, things that can't be engulfed. They do so by releasing chemicals that are going to destroy it. They do so cytotoxically. So their role then is as a cytotoxic white blood cell. Keep those in mind as we talk about the white blood cells. What is their main role? What are they doing? We also mentioned the mononuclear phagocytes, the macrophages, if you will. Main role is as a phagocyte. If they're in tissues, we call them macrophages. If they're circulating in the bloodstream, we call them monocytes. And then we introduced a third type of macrophage, a dendritic cell. And dendritic cells, they're kind of collectors, if you will, collectors of foreign material, foreign products, bacterial cells, things like that, for presentation to T cells. And we'll talk more about that here shortly as well. And then we briefly introduced the lymphocytes. Those were the B cells and the T cells, and they were part of the adaptive immune response. They are part of the learning memory immune response that we have. We haven't got there, that'll be chapter 16. But we did introduce them as well. And then we talked a little bit about cell communication. We mentioned surface receptors and cytokines. Surface receptors being the proteins that are on the surface of the cells, and cytokines being the chemicals that they release. And this is how the cells are going to relate back and forth to each other, what's going on in the body, what needs to happen. Both are going to be releasing chemicals, and they're going to have surface receptors that will bind the chemicals being released by other cells. They may have surface receptors that are bound by the chemicals that they are releasing, so they can release chemicals that affect themselves as well. But all cell communication essentially is going to be done through this process of cell receptors and chemicals that are being released by cells. Back and forth, letting the cells know what's going on. We mentioned adhesion molecules briefly in this cell communication scenario here. I would consider them kind of to fall under the category of surface receptors, because they are molecules on the surface of the cells that attach to other cells and slow them down so that they can move into the tissues. This would happen in the case of an inflammatory response. That brings us now then to sensor systems. Well, sensor systems are going to involve surface proteins and proteins that are going to be circulating in the bloodstream. As far as surface proteins, there's a special type called toll-like receptors. That we will find on most cells, if you will. Many cells have toll-like receptors. Macrophages, we can talk about them having them specifically, but toll-like receptors are surface receptors that recognize specific bacterial molecules. Remember, we talked about the innate immune system being non-specific, but in this sense, there's some specificity put into the innate immune system because the toll-like receptors are specific bacterial. They're responding to bacterial molecules like peptidoglycan, like lipopolysaccharide, like flagellin. Those are uniquely bacterial molecules. And so if we have surface receptors that can recognize those, it can then serve to set off an alarm if you recognize enough of the right surface receptors. So if you imagine our macrophage or some other cell maybe has several of these surface receptors for lipopolysaccharide and for peptidoglycan, and maybe it takes 2 or 3 different surface receptors to be bound before the cell now starts to release cytokines saying, hey, something's going on here, there's bacteria molecules in the neighborhood. Usually triggers some kind of a response, maybe an inflammatory response, maybe it draws neutrophils to the area, maybe it draws macrophages to the area. In addition to toll-like receptors on the surface, there are also receptors inside called NOD proteins. I won't get into what NOD stands for because we don't really need to know that. But they're much like toll-like receptors in that they're proteins, if you will, inside the cell. I should put a nucleus here. But these proteins inside the cell will bind to bacterial products, bacterial molecules and things, and again, set off the appropriate response. So toll-like receptors sample external molecules. NOD proteins sample internal molecules. It's a way of determining if you've been infected, if you're sampling internal molecules. One other type of sensor system, complement. This system here consists of a bunch of different proteins that are circulating in the blood. And these proteins, for the most part, are inactive. But they can serve to augment the activities of antibodies. So what happens is that antibodies can serve to turn on the complement system, and then you can get a cascade of complement proteins being activated. Generally they're inactive, but when one becomes activated, this cascade can trigger the activation of other complement proteins. And what does that then cause to happen? Well, a number of things. Could lead to inflammation. We'll talk about inflammation here shortly, but one of the things that complement does is it serves to attract macrophages to an area, sort of chemotaxis. Lysis of foreign cells. So bacterial cells, complement can form pores, if you will, holes in the membrane of bacteria cells. You form enough holes in the membrane of bacteria cells, they take on water and oil, and then they burst. Bacteria cell dead. opsonization. This is a process by which the bacteria become coated with complement proteins. So what happens now is that the bacteria are coated with complement proteins, and then the macrophages can recognize the complement, engulf the bacteria, remove it from the circulatory system, or the tissues, or wherever it might be. Any questions about these two sensor systems? Yes?
You said something about cytokines?
Cytokines, those are part of the communication system. Those are the chemicals that are being released that will bind to surface receptors.
So those are the blue and purple?
They're not in this discussion here, but if we take this cell, for instance, and we put surface receptors on it, and then another cell was releasing chemicals, these chemicals would bind to these surface receptors if they fit. These are the cytokines. Any chemical that the cell is releasing could be technically a cytokine. It's just a messenger. It's a chemical messenger to neighboring cells or to other cells. And the way it communicates is by releasing a chemical. Now, the cytokines can vary with what their role is. Now, some of them can cause a cell to start to divide. Some of them can cause the death of a cell. So it really depends on the cytokine that's being released, the cell that it's binding to. So there's some specificity there with regards to the cell surface and the cytokines. If you don't have the surface receptors for any particular cytokine, then that cytokine does not affect you. You don't respond to it. One of the important roles in our innate immune system, mainly for the macrophages, but the neutrophils also, and that is phagocytosis. So I thought we'd take a minute and talk about phagocytosis. You know, generally it's the engulfment and digesting of material. The macrophage is going to recognize certain things on the bacterial cell, either complement proteins or antibodies, or maybe there's some other pattern that the phagocyte recognizes, like toll-like receptors. This could then cause phagocytosis to occur. Other things that can happen if cells have been damaged or there's other chemicals being released, you could have your complement system turned on, and so you can have a cascade of complement here. And one of the things with that is the release or into the chemical environment, complement 5. Complement 5 kind of serves as a chemical for chemotaxis, and it's going to bring our macrophage to the area. So whatever's going on here, damaged tissue, infection, something else like that, cytokines or antibodies have caused the complement cascade to begin. So now you've got C5 being activated, and it's serving as a attractant to bring the macrophages to this area where maybe the infection is going on. In addition to that, part of this cascade is that you have a complement, C3, that will bind to bacteria cells now. This complement has been activated and it starts to opsonicize the bacteria. This now has been marked so that the macrophage can recognize it and engulf it. The complement kind of serves as that marker for the macrophage. The macrophage has on its surface, of course, receptors that will be specific for this complement. So now it doesn't really have to have different receptors for every type of bacterium that it could come into contact with. It only has to have receptors for complement. If complement coats the bacterium, it can recognize the complement and then engulf the bacteria. So now it sends out its pseudopods. We engulf the bacterium. So, keep my list going here. We have chemotaxis has occurred. We've had recognition and attachment. And now we have engulfment. With engulfment, we get the formation of a phagosome. So this is a vacuole, pretty much, that's got the bacteria in it. Out here in the cell are little packets of digestive enzymes. And these little packets of the digestive enzymes, they're going to fuse with this phagosome, and they're going to release their materials into the phagosome, the digestive enzymes, and these digestive enzymes then will serve to break down the bacterium